tube number 1? An inoculating loop. What would you use for test tube number 2? An inoculating loop. Surprise, surprise. What's the technique that you'd use for this one? I don't know, a dip and swirl is what we call that. What do you use for a slant? We call it a fishtail across the surface, okay? All right, what is the tool and technique used for, this is for tube 1 and for tube 2, so you guys have already covered this, right? I know that you guys look at that and say, man, that's easy. This is almost the most missed question on that exam. Okay? Please do not miss that. It is easy.
So we can say dip and swirl for number 1?
Sure. But you have to say the inoculation tool also. Most people gave me just half an answer for this. They would say inoculating loop and forget about the second part of the question. Okay? It's not just the tool, but it's the tool and the inoculating technique. Fair enough? So read the question. Okay, what is the gram reaction you see right there? Gram positive. What is the shape? Can't just say positive. It's gram reaction and shape. Those are not rods. Those are coccus. A gram positive coccus. What is the purpose of iodine in a gram stain? It is the mordant. That's exactly what I want you to put down. Okay? Failure to add alcohol during gram stain would do what?
It won't be.
Everything would be purple.
Yeah.
So you couldn't differentiate gram positive versus gram negative. You with me? Mm-hmm. Okay. Why is the gram stain not work on mycoplasma?
Because it's a wax.
What is the gram stain supposed to point out? The cell wall. Does mycoplasma have one?
No.
So, mycoplasma has no cell wall, it won't work. Okay, that's what I'm looking for.
You need heat.
Huh?
You need heat.
Well, you need a completely different staining technique for that one. It wouldn't work at all. Alright, you guys with me? Alright. So, this media right here would be used for what? I know it's a different color, it got a bad transfer. Usually for us, it's green at the top. Y'all remember that one? So what is it used to identify? Y'all don't remember that green tube? That was TGB. Bioglycolate broth. You're right. It is supposed to measure whether or not you've got an aerobic versus anaerobic bacteria. The green at the top is supposed to show you how far air goes down into the tube. Okay? So if it just grows down here where oxygen can't get, it's anaerobic. If it grows up here at the top, it's aerobic. And if it grows all over, it's facultative. Okay? So second question on this. How would you classify an organism if it only grows in the fluid below the red band on this one? Anaerobic. If it grows away from oxygen, it's anaerobic. If I said, what if it grows just at in the red area? It's aerobic. Okay? Are these questions fair? Yes. Are you guys going to read these and say, man, I can't remember anything? Yes. Okay. To prevent refraction from the 100x objective, what do you have to add to your slide?
Oil.
Immersion oil. Perfect. Okay, why do we use a basic dye to perform a simple stain? We just talked about this. Bacteria have a negative charge. Acidic dyes have a negative charge. You need basic dyes which have a positive charge so that they'll stain the bacteria. Okay? If you try to do this in like a couple of words, you say, why is a basic dye used? Because it sticks to the bacteria. Okay? That's really the best answer. It sticks where acidic dye would not stick. Fair enough?
Mm-hmm.
All right. You guys don't have to worry about E and B. We haven't got there yet. Or this one. Okay, to prevent the spread of contamination, what kind of techniques do you use? Aseptic. Fine. Okay. What's the minimum of time you ought to apply disinfectant using paper towels to every spill that you make? This is in your lab manual. Okay? You need to know this. It should be in our safety sheet at the very beginning of this. Okay? The safety sheet that we went over, when you have a spill in microbiology, you have a certain amount of minutes you are supposed to apply disinfectant with paper towels over it.
1 minute?
20 minutes.
Are you reading it? It is 20 minutes. Okay? You guys with me? That is in your safety sheet. I know you're sitting there thinking, man, I didn't think the safety sheet was going to be on the midterm. Oh yeah, safety sheet works its way on there. Okay, you don't have to worry about PEA. We don't do this one, and we don't do catalase yet. This one we do. We have done. What are the green structures on this slide? Those are spores. Don't they look pretty? I did the spore stain. I know. I'm awful proud of it. Okay. What's the question is, name 2 genera of bacteria that we study that produce spores. Okay, so if we talk about genera, okay, this is not species, this is just general bacteria. So what are 2 that we've dealt with that produce spores? You're close. Okay, Clostridium was one that produces spores. And what's the one we did today? Okay, what's the first word? It's B-megas bacillus megabacterium. Megabacterium is the species. You don't have to worry about it. All you'd have to do for this is say bacillus or Clostridium. Okay? Those are the only 2 spore producers we know. So it's either bacillus or Clostridium. Either one's right. So if it's asking for genera, those are the genuses. Huh? No. I've never counted for spelling. So, these are spore-producing bacteria. We only deal with 2. We deal with Clostridium sporangiens, which you just have to say Clostridium, or we did B-meg, which is Bacillus megabacterium. For this, all you have to do is give the genus. So it's either Bacillus or Clostridium. That's all you have to put.
Or B-meg.
If you wanted, I'd take it. That's more information than I'm asking for. All you'd have to do is give me Bacillus, and that's enough. Okay. Prior to using media, they get sterilized using what? Autoclave, 30 minutes. What is an ingredient added to the media that makes it solid? This is in one of those background information pieces.
Isn't that powder stuff we
It
use?
is, but what's it called?
That was many moons ago.
I know, but I'm telling you, this is all the information that we've gone over for the first half of the semester. Y'all remember something called agar? Where does it come from?
Algae.
Red algae. Okay, that's important. Agar is what makes it solid. Otherwise, it's liquid media. You absolutely need to know that. I would know that the red algae produced agar. Okay?
Is it agar or red
Agar.
algae?
Agar is the thing that you add. That's the ingredient. Red algae is what produces agar. You guys with me? Okay. What else do we have here? You don't have to worry about this one. Don't have to worry about this one either. Man. Okay. What kind of organism is this? Huh? This is not endospore. This is a really big one. You see how it's got all these cilia on it? It's got cilia.
Oh.
You have three choices. Paramecium is right. Paramecium has got cilia, Euglena has a flagella, and the amoeba doesn't have any. You guys with me? So you see cilia, that's paramecium. Okay? Now, I don't want genus or species. All I want you to do is tell me generally what we're dealing with. Okay? Somebody told me protist. I probably would have taken protist. Okay? But this is paramecium. What is one feature that this cell shares with prokaryotic bacteria? What's that? Well, it could have cilia. I mean, you could have told me 100 different things. You could have said plasma membrane. Huh? They don't have cell walls. No, it's fine. So plasma membrane, they both have plasma membrane. You could say they both have respiration. There's tons of things you could give me. I'll take any of them. It just has to be true. Okay? That question is generally just asking you what things do
Like our last
prokaryotic
class,
bacteria have in similar to eukaryotes. Anything that we can come up with is true. Okay, we already talked about this. 37 and 25 degrees. What does each represent? Body temperature and room temperature. Okay. How do we store our Petri dishes? Upside down. Why is that? It's just the way you do it. I love that answer, but it's not exactly what I'm looking for. Have you guys ever gotten a cold drink out of the fridge and taken it outside? What happens? You get lots of condensation, right?
Yeah.
That's the same thing that happens within your plates. It almost always gets condensation on the lid. Well, if you get condensation on the lid, it can drop onto your sample, right? Which would add contamination. So we store them upside down, so when you get condensation, it can't drop up, right? So it's stuck on the lid. That's why we store them upside down, so it doesn't get contamination. All right, so why did we use nigrosin for this stain? What's that? Well, you do see the background, but that's not why you'd want to do it. What is the purpose of a negative stain? I know we went over this. Why do we do negative stains? What do you get to see? Yeah. You do a negative stain so that you can see the true shape of bacteria. That is the only reason. Okay? Because you don't have to heat fix it. Okay? You guys with me? So if you see a negative stain, the only reason you do that is to see the shape of it. Okay? What's the shape that you see? Those are rocks. So that's exactly right. Okay, you don't have to worry about this one or this one. We haven't done the acid fast stain yet, so you don't have to worry about it. And we've got some stuff to pick up, but these three you don't have to worry about it. Alright, we did do this one. What are we looking at? It is not BMEG. We did a slide and we looked at this. I think it was the second lab. Might have been the first one. What does this look like? What's that? Huh? It looks kind of stringy, right?